Hello, everyone. My name is Menachem Oshelion. I'm a researcher at the Hebrew University of Jerusalem at the Faculty of Agriculture. I'm a plant physiologist, and my main research is to study the plant-environment interaction, in particular crop plants, and in particular I'm studying stresses, nutrients, and today I want to talk to you about a research we did of studying biostimulant and a novel telemetric system that we have here, so we can make a physiological phenotyping, or as we call it, functional phenotyping. So first, few words about what is biostimulants. So biostimulants are actually organic materials which per se do not help the nutrition of the plant, but enhance the nutrient efficiency. In particular, this substance helps the plant abiotic stress tolerance and general vigor. Nevertheless, to develop a biostimulant takes a lot of time and energy because there are many groups of efficient biostimulants like amino acids, humic acid, fluvic acid. Many people know seaweed extract, inorganic component, and so on. These are organic matters, and some people also use living bacteria like fungus or bacteria that also can benefit the plant growth. But the general concept of all of these materials that you need to use a very small amounts of these materials because it just stimulate the growth and the resistance or tolerance of the plant. Now, when you want to develop a new substance like this, it's really time consuming and expensive because you have many options, many products that are possible to work with, but it's hard to know is it going to work or not. And doing a field trial takes a lot of time, a lot of effort, and you also have a lot of noise. Now, in the coming years, the EU regulations will require standardization of these processes. So you will have to be able to provide a very clear-cut results and very standardized way of working. A normal development of biostimulants, even the simplest one, can take 2 to 4 years. It's not as long as to breed a crop, but it's a lot of time and money. And the main bottleneck is the phenotyping process. So to know whether your material works, have some beneficial trait, or does not. So this phenotyping today at the pre-field stage, and it's important to mention, we talk about the pre-field stage because we don't want our plants to go to the field each time we want to study new material. It will take a lot of time, a lot of money. Most of this material does not work. So this phenotyping is like this. If you take all the material and want to know which work, is like this gambling which horse is going to win the race, right? All of them should work. You have a good rationale to support each and one of them. But in the end, to test the winning horse, you actually have to test them in a real life in the field. So this is why we are using functional phenotyping system, which I will elaborate soon. In the study we tested, that I'm going to report now, we tested two kind of very famous biostimulants. One of them is amino acids, used a lot in this field. And another one is a seaweed extract. I think this is the #1 biostimulant in the world today. We wanted to test their capabilities based on a functional phenotyping. The experiment involved pepper plants, and we wanted to measure all the plants simultaneously. So we know that the plants see the same light, same temperature, everything the same. Some of them will have the seaweed extract. Some of them will have what we call the NUFO, which is amino acid cocktail and polysugars. Both of them are commercial products. We wanted to test this treatment under well-irrigated condition and under drought. So all in all, we have 6 different treatments. Each color here represents a different treatment. And we want these treatments to be, as I said, simultaneously. Now, when you do phenotyping, you have to ask yourself several questions. First, what do you want to phenotype? The plant structure, function, length, size, what? How to phenotype? How you want to do it? When you want to do it? Where? In the greenhouse, in the field? And one of the main challenges is how to translate the data you get, and you get a lot of data using phenotype, to practical decisions. So if we go with the first question, what to phenotype? So mainly when we talk about stresses, we talk about plant-environment interaction, which names as G times Z, genotype stamp environment interactions. Now we must remember that the plant sees at a single moment the atmosphere, light, temperature, VPD, humidity. This atmosphere impacts the shoot, and the shoot actually impacts the root, and the root impacts the water in the soil, and the water in the soil impacts the rhizosphere, all the bacteria and fungus in the soil. So all in all, we have multiple interactions which all the time cross-interact with one another. So in order to really to do a good phenotype, you have to measure the soil, the root, the shoot, and the atmosphere simultaneously and continuously. Okay, this is a very dynamic system. So the plant takes the water from the soil, transpire it from the guard cells, and losing most of the water in order to gain some CO2 back. The ratio between this water loss and CO2 gain is very bad in crop plants. It's about 500 to 1. Now, we want to know whether our biostimulants are going to improve this ratio, in particular on the drought. So for that, we actually testing this functional phenotype. So now we're talking about how to phenotype. So we wanted to phenotype using a lysimeter, a gravimetric system. This is a well-known way to measure plants' physiological parameters. Only we did not want to use this old scales as it's in the picture, because this lysimeter has a lot of problems. The technology is like 40 years old, connected to data loggers. You have a lot of problems, especially pot-related problems. I don't want to elaborate, so we publish this in this paper, all this problem. So we use a much more modern metric system of a company called Plant-DiTech, which actually help us doing the functional phenotyping using all kind of plastics and a controller. This system actually control the soil water, measure the atmospheric condition, and allows you to do a feedback irrigation for each plant separately. So the system actually measures for each plant in the array the soil, the plant, and the atmosphere in one continuum, and the system do it every 3 minutes. So we have a continuous measurement of the plant water balance. Now the system has a controller with 2 valves, so we can make any cocktail we want. For example, with the biostimulant, we can make any concentration to each one of the plants at each time of the day. And this system actually replaces the old data logging system and also solves many of the problems with pot. When you do experiment with pot, especially drought, you have a lot of what we call pot effect. So as I told you before, this paper here we published actually explain how the system solved this problem, so I'm not going to go about it. The third question is when to do the phenotyping. Now the system here also solves you this problem too, because this system continuously and simultaneously measure all the plants in the system simultaneously. So if this is the time of the day, the x-axis, and the y is the whole plant transpiration, we actually have a continuous comparison of all the plants all the day. So in the morning when there is no different, I will see no difference, but at noon I will see the difference. So again, when you measure plant-environment interaction, it's very important to have a continuous and simultaneous measurement, which means all the plants should be measured simultaneously, so we know each one of the plant sees the same environment as its other plant, its neighboring plant. So what do we measure all in all? We measure the whole plant biomass gain. So this system giving us the plant biomass gain, the true biomass, the fresh weight, continuously every day, the plant transpiration, the plant water use efficiency, stomatal conductance, and root fluxes. All of these are very core physiological traits, which really enable you to do the physiological or the functional trait based on the plant response to the biostimulants, and plant response to either biostimulants and drought treatment, just the drought, or just the biostimulant. In addition, we also measure many other conditions like the soil temperature, water content, light, VPD, and so on. The system also enables us to do real statistics online, so we can look on the raw data, we can have averages, we can click on a button and we get a t-test or ANOVA Tukey test on all the treatments, so we get also the significant, and everything is online. This would help us saving a lot of time, and more important, translating the data to knowledge. As I told you before, you get a lot of data from this system, so if you have a tool that enables you to get the data online in real time, you can really save a lot of time by regulating your experiment or controlling your experiment. So there are many functions, I'm not going to get into it, also histograms, a heat map of each plant in the area, and so on. So again, the system enables me to control the treatment for each pot separately, both the biostimulant and the drought, and do combination of the treatment for each plant. Just to remind you, so I had 6 different treatment, control plus, well rated plus the biostimulant, each one of them. So blue, green, and orange are the non-drought with the different cocktails of biostimulant, and the same biostimulants under drought, which is the hollow circles. We used normal pepper fertigation, only we dilute it in 20% because we really want to check the nutrient efficiency. So we didn't want to give the plants the best condition, but to put them under slight nutrient stress. This is why we dilute a bit the concentration. The phenotyping was done by growing the plant under optimal conditions, then exposing them to drought, checking at which point the stomatal start to reduce. This point is very important because this point is actually when the drought starts. We call it the theta creep. We measure the rate of closing the stomata, and then we recover the plant to measure how fast and how good the plant recover. Actually, the delta between the point the plant recover and the control is the penalty of the treatment, the drought in this case. And of course, we compare all the treatments simultaneously. Again, the optimal condition, the drought conditions, where we measure the theta creep, the point where the plant senses the drought, and the recovery. We use mild to moderate stress because we want to mimic stress that many times this pepper sees at the farmer greenhouses. And we did this functional phenotyping on this. This is how the raw data look. On the top, we have the environment, soil condition, light, and VPD in a continuous manner. Each color is different treatment. So these plants are the one that was not exposed to drought. We have the seaweed and the fall one treatment, and the same only with drought. Since the system giving us the plant weight continuously, we wanted to check the dry weight of the plant taking in the end of the experiment with the plant calculations. And we got a very nice correlation both in the dry treatment and the normal treatment. We did the same also with the shoot fresh weight and the dry shoot weight. Both of them gave us a very nice correlation. So we saw that the data is very reliable. So based on this, we look on the plant's behavior. So this is we did the experiment in 1 month. So I forgot to tell you, all of this scenario take about 30 days, okay? And what we see here, so here is where the experiment starts, here in day 1. You can see that the daily transpiration of all the treatment was the same. It took some time, and then we start to see that the seaweed really improved the plant transpiration. You can see the green line. After it was the FO1 treatment, and then the blue is the control. Where we expose them to drought, we can see that, interestingly, the seaweed actually was the first to dry out. This is the green line here. So exposing the plant to drought really show us that the plant that behaved the best under normal condition actually did not perform that good under drought. You can also see the soil water content of the seaweed, which is just dry faster, which is a not good sign when you expose the plant to drought. Another interesting thing is this tetacrit was not different between the plant, but the slope of dryness was slower for the FO1 treatment, which means it maintained the water better than the other plants. Okay, so although it has significant more transpiration than the control, which is a good sign because the transpiration is actually linearly related to photosynthesis, it still maintained the water very nicely in the soil. We also check the plant growth rate. Again, we see similar pattern to what we see in transpiration. And when we take the shoot dry weight, we get actually similar pattern. So the seaweed increased the biomass the highest, then the FO1, the FO1, and then the control plants. Under drought, we see no difference in all of the treatment. When we normalize the transpiration we saw here to the plant biomass, we actually get that the plant transpire less per gram. So the plant lose more water, but less per gram plant because they were bigger, which means they have a better water use efficiency, which is very nice. So we can see that this treatment really improved the water use efficiency of the plant. Next, we want to check how the treatment improved the resilience or the recovery rate. So resilience is how fast the plant recover after stress. So this is the stress, what you see here. And here is the recovery rate. We measure recovery in two ways. First, how fast the plant go back to its previous stomatal conductance or transpiration. We see here that the seaweed was not performing so well. Actually, it was lower than the control. But the FO1 treatment not only recover as the control, it also gain water. Its water reabsorption much better than all the other treatment. So in fact, this treatment not only improved the transpiration and the growth, but also improved the recovery rate after stress, which is very important trait, both under the water reabsorption under normal condition and also when you normalize the water reabsorption, which means how fast the plant get the water when you give it to it after the stress. You can see that when even you normalize to the plant size, it's still significantly better than the other treatment. Taking the yield, so we saw that the seaweed under normal condition really improved the yield, but under stress, it was just collapsing. The seaweed did not, the other treatment did not show significant improvement in yield, but I must remind you, this is only 30 days, so it's maybe not enough time, and maybe this will be even better after 3 to 4 months, the normal time for yield. Interesting, we saw a very nice correlation between the transpiration and the plants and the yield. Actually, today we use this correlation to select the plants even before the trait. We take this transpiration, which is very easy for us to measure on the system, and using this transpiration as a physiological trait, we can actually select in a very nice correlation to the plant which will perform better. Looking at the root, we find no difference not in the root flux or the root biomass. You can see very similar results. And all in all, we conclude the data in this way. What we saw is that under normal condition before the stress start, at this point, it's phase 1, the seaweed actually improved the plant vigorous growth. Nevertheless, this fast growth make it more susceptible to drought because it dried very fast and recover even a bit slower. While the FO treatment improved a bit the growth under phase 1, but improved the drought response significantly and also improved the resilience. So the plant recovered faster, which is a very interesting result because it means that this mechanism of action is very interesting for a post-stress phenomenon. So to summarize our result, we saw that the whole plant soil atmosphere continuum, the SPAC, is a very good functional trait which lead to a very nice and strict result that repeat themselves. Using the Plantarray system, we can control each pot separately because I want to remind you that the system control each plant separately. So we have a full control both in the irrigation, the application of the treatment, and the measurement on each plant separately. So we can really build a randomized structure. Each plant really get the treatment where we want it. Doesn't matter where we want to put it. We measure many physiological traits which under drought condition are very important. We measure continuously and simultaneously, so we get millions of data points, but because we have a very nice software, we can analyze it on the spot. And actually, because we finished the experiment in 1 month, we can push in one season 3 consecutive experiments. So we have standardization and repeatable condition. So all in all, we could short down the development of a new biostimulant from 2 to 4 years to about 1 year, which is a significant improvement of this method that I just explained. We publish this data, so if you want to see more details about this technique and the research, we publish it. The research was funded by ICL, a fertilizer company, and published in Frontiers. If you have more questions, or if you want to collaborate or ask me anything about this particular question or any other physiological related or functional phenotyping, please feel free to contact me. Thank you.